Assalamualaikum to respectful examiner Dr. Jeffrey Jaafar and fellow friends. Hi, my name is Arina binti Hamdan and I am going to present on my research project titled Epiphytic Species Algae as the Revival Potential of Feedstock Collection Case Study. I am supervised by Dr. Engineer Amizun Azizan. Now let us see on my abstract. Microalgae as a biofuels are a perfect substitute for liquid fossil fuels, since microalgae have a significant ability to convert atmospheric carbon dioxide to useful products such as carbohydrate, lipids, and other bioactive metabolites. In this research, two case studies are conducted, where case study 1 is to study on the growth possibility of triboside species on media such as Algae agar, yeast extract, and algae broth. As for case study 2, it is about reviewing on the biofuel production from microalgae techniques, the potential species of epiphytic algae and its other application, and species of microalgae that is high in biomass composition. So let us take a look for the introduction, shall we? Okay, algae has been recognized as potentially good sources for biofuel production because of its relatively high oil content and rapid biomass production. These types of green algae can be found mostly in freshwater. However, in this research, the green algae sample is taken from epiphytic terrestrial species. That it make this research project is different from the previous research project. Okay, case study 1 is about studying the potential growth of triboside species, which is a type of epiphytic algae that are collected from Areca catechu that is located at Complex Kejuruteraan Tunku Abdul Halim Muazzam Shah, UiTM Shah Alam. And then the growth was conducted on a media such as LB agar, yeast extract, and LB broth. And the growth of the culture algae were observed. As for case study 2, it is on reviewing the biofuel production from the microalgae species technique, the potential species of epiphytic algae and its other applications, and species of microalgae that are high in biomass composition. The microalgae technique includes stages such as cultivation, harvesting, extraction, and transesterification. Next is the three species of epiphytic algae reviewed are Phycopeltis algae, Triboside species algae, and Trentifolia algae. On top of that, the Sedismus obsticulus has the highest lipid contents, which are 52%, followed by Butyrococcus brownie with 36.9% of lipid contents. So why don't we continue with the objective for this research project? So the objective of this research project is to study the growth possibility of epiphytic algae on media such as LB agar, yeast extract, and LB broth. The second objective is to review on the potential species of epiphytic algae and its other application, and species of microalgae that are high in biomass composition. So there are two problem statements for this research project. The first one is, can the epiphytic green algae species grow on the proposed media such as LB agar, yeast extract, and LB broth? Next is, does the review on the epiphytic species algae biomass producers be used to produce biofuel or maybe other application? So why don't we go through case study 1? Okay, here are the case study 1. So let us take a look on the methodology for case study 1. At first, the collection of epiphytic algae from Areca catechu, it is by using a forceps and then stored in an aperitif tube that contain formaldehyde for preservation, and then it is stored in a refrigerator until the further use. Next, in a lab, we prepared LB agar, LB broth, and also yeast extract. And next is the cultivation of algae, where LB broth, LB broth will be put in an incubator shaker at 250 rpm for 27 degrees Celsius, while for the LB agar that's stored in a petri dish will be stored in a refrigerator. Next is a daily sample taking for the cultivation of algae and then stored in a freezer for further observation. Next is to observe the growing of algae colonies. Let us take a look on the result. So result. Okay, so here's the result for the LB agar. In day 1, there is no colonies formed. And for day 9, after day 9, there is also no colonies formed. And so does on day 15. As for yeast extract, in day 1, there is also no colonies formed. Same goes to day 9 and day 15. Okay, next is the LB broth. For LB broth, in day 1, there is also no colonies. As for day 9, the color turns from clear to cloudy, and also the smell is like a pungent smell. And for day 15, there is a colonies or like a small ball like in the conical flask. Okay, so let us take a look for case study 2. Okay, for case study 2, processing of microalgae strain into microalgae biofuel. The first stages is cultivation, where it is run under controlled conditions to obtain valuable end products. Next is harvesting, where the algae harvesting can be recovered biomass recovery from the total cost of production. Next is the extraction, where the extraction of lipids in the algae occur. And lastly is the trans-esterification, where the reaction to produce simple ester, which is better known as biofuel. Next is the epiphytic species algae that might be potential. Firstly, epiphytic means it is not parasite, but it grows independently without taking up nutrition from the host plant. Okay, here are some examples of epiphytic algae species that are being reviewed. Okay, the first one is phycophytic algae. Okay, as you can see here, this is the morphological of the algae. The algae can grow on the leaf, on the twigs, fruits, and stem at the higher living plants. Okay, for the figure 11, it is a Triboxia species algae. Most commonly recognized species rich group of terrestrial lichenized green algae. Okay, lichen. What are lichen? Lichen are a type of symbiont between fungi and algae, where the algae will produce products such as sugar through photosynthesis process. Then it will transfer the sugar to the fungi, and the fungi will use it to produce lipids. As a symbiont, the algae will then obtain dissolved minerals and nutrients and water from the thalline surface through the cell wall of fungi. According to Asmida, Triboxia species can be found in Baritonia resmosa, and this type of algae is used for biomarker. In her journal, it says that the algae growth rate is said to be increased where in the area with a high carbon dioxide. Okay, next. On figure 11, as you can see here, is a Trentifolia algae found on a rock of Mount Gonga. Okay, Trentifolia is known as cortical species of algae, meaning that it grows on barks of woods or the deciduous tree and conifers. And this type of algae is a bit different from typical algae. It is because of it has a red, yellow, or brown color because of it contains carotenoid in the cell. Okay, therefore, based on these three epiphytic species algae that I've been reviewed, it can be seen that it has other application, such as one of it is biomarker. So I think further research on the epiphytic species algae for biofuel might be beneficial since the availability of the species are accessible. Okay, next let us see on the energy storage composition. Okay, here is a graph that shows on the species of microalgae that are high in biomass composition. Okay, from the graph, we can see that Syndesmus obtuscus has a higher lipid content compared to the other six species of microalgae found. So, since it has higher lipid content, it is beneficial for the algae to produce biofuel because of its higher lipid content. Okay, next we continue. Conclusion. Conclusion. Okay, lastly, I can conclude that in the growing algae sample in case study 1 should be investigated to find out the potential of Chiboxia species on the algae broth since it has a positive growth on the algae broth compared to the agar. Again, next, for case study 2, epiphytic species algae that are reviewed in this paper are believed can be a suitable feedstock due to their availability. The third generation of biofuel in the future may play a vital role in fulfilling global energy demand. However, the current harvesting process techniques are expensive. Therefore, more research and investigation should be given to execute a cost-effective production process, including achieving a higher yield. And therefore, thank you.